Dear students, Assalamu alaikum to all of you in botany class. Our today's topic is virus and viroids. First of all, we have to see what are viruses. So, this term at first was coined by Pasteur in 1880. Pasteur told that these viruses are not living. These are truly living. These are not truly living. It means that when viruses are not truly living, when they enter the body of an organism, they become active and they are living. They start replicate and start division for the to harm the organism. And when they are outside the host, they are non-active or they are non-living. So, as per Pasteur's concept, we know that viruses are infectious organisms which has not any proper cell and they are obligate parasites. It means that when they have dual nature, when they are outside the body, they are non-living. When they enter the body of an organism, they start to replicate, or we can say they start division and to harm the host. So viruses are not truly living. Then another man came. His name is DJ Ivanovsky in 1892. He told that these viruses are smaller than bacteria. On his observation that he has taken the viruses in and he told that viruses because viruses pass through bacteria proof filter. Then another man came and he told his name is M.W. Bajering in 1898. He told that these viruses cause infection in their surroundings. They release an infectious fluid which we call as contagium vivum fluidium. It is a living infectious fluid. Then W.M. Stanley came in 1935. He told that viruses are crystalline proteins. These are in the form of crystals. So he also told that these are obligate parasites. Now the important point here is that first of all, as per Pasteur's concept, we know that he told viruses are not truly living. On the basis of which he told, it means that they have dual nature. They are neither living and they are nor non-living. So I have told you that when they are inside the host, they are living, and when they are outside the host, these are non-living. And he also told that these are as per this he told that these are non-cellular infectious organisms. When we see what are the characters of these viruses, we know that these viruses infect all types of living forms, plants, animals, etc. And these are obligate intracellular parasites. They multiply inside the host, and these are observed under electron microscope. Viruses are 10 to 100 times smaller than bacteria, and their size ranges from 20 to 300 nanometers. Then these we can say these viruses can easily pass through bacteriological filters which are smaller than bacteria. Size of virus is measured by three methods. First one is ultracentrifugation. Ultracentrifugation. First one is ultrafiltration. Second one is ultracentrifugation, and third one is electron microscopy. these do not have any kind of cellular organization, and the most important point is that they contain DNA or RNA as genetic material, but not both the materials at the same time. So these viruses look like the essential enzymes for protein and nucleic acid synthesis because they lack the required essential enzymes. And we can say these viruses depend on host cell for its replication and multiplication, and these viruses are also unaffected by antibiotics. So virus is a great concern in the field of microbiology because these are responsible for causing human diseases like AIDS, common cold, smallpox, etc. And these viruses are responsible for cancer in birds and animals also. Viruses may be enveloped or non-enveloped in nature. Envelope is made of lipoproteins, and these lipoproteins are present in the host cell membrane. Then these viruses require a living media like embryonic egg or cell culture. And these viruses are of two types: DNA virus, which means deoxyviruses, and RNA viruses having ribonucleus as ribonucleus as RNA virus. Riboviruses having RNA as genetic material. Each virus having two types of double stranded and single stranded genetic materials. So we can say they have genetic material present either DNA or RNA, but their genetic material cannot be um, cannot be both at same time. So viruses have genetic material DNA or RNA. These viruses are infectious proteins, or we can say these are nucleoproteins because these are nucleic acids, and we can say these are infectious in nature. When we talk of DNA or RNA, we know that these are nucleic acids. So, as far plants are concerned, these viruses cause harm plants. Harm plants. The viruses which harm plants have genetic material single-stranded RNA, ssRNA. The viruses which harm animals have genetic material ssRNA or dsRNA and dsDNA. The viruses which feed upon bacteria are called bacteriophages, and their genetic materials dsDNA. Now, one bacteriophage is 5174. This has genetic material ssRNA. Now, the important point here is that viruses have a protein coat which is called as capsid. This capsid is made of smaller units. These units are called capsomeres. So, we can say genetic material of viruses is surrounded by a protein coat which is called a capsid. So, viruses have high molecular weight as compared to viroids, which are smaller than viruses. Some categories of viruses are here. First one is adenovirus. Adenovirus is the virus which causes lung problems, and then there is rhinovirus. Rhinovirus is the virus by which nasal infections, which is responsible for nasal infection, for example common cold. Then there is retrovirus. Retrovirus is responsible for human immunodeficiency virus, and its genetic material is RNA. Then we have to see what is the effect of virus. What are the diseases which are caused by viruses? So in case of animals, we know this virus is responsible for AIDS, mumps, smallpox, influenza, etc. But when we talk of plants, in case of plants, these viruses cause diseases like leaf rolling, leaf curling, leaf coloring or yellowish color of leaves or mosaic information, for example in case of tobacco mosaic virus. So we have to remember here from examination point of view the structure of TMV and it is bacteriophage. Then if anybody will told us that what is difference between virus and viroids, so we can say viroids are smaller than viruses, and this term viroid is discovered by T.O. Daniel in 1971. And these viruses have low molecular weight, but these viroids have low molecular weight, but in case of viruses they have high molecular weight. And we can say viroids have free RNA only, and these cause potato spindle diseases. These lack protein coat. So there is another term which is prions, but in case of viruses we have protein coat, but in case of viroids we have no protein coat. There is another term which is prions. Prions are the free molecules which are non-cellular and create brain related problems. So we can say these are abnormal proteins, and these abnormal proteins, these cause misfolding of proteins. That is, for example, bovine spongiform encephalopathy, BSC. It is called mad cow disease. This mad cow disease is present in the brain of cattle. In humans it can cause C or Jacob encephalopathy, uh, JN C or Jacob encephalopathy. So this causes membrane loss, or it can cause neuron damage, it can cause headache, and it can cause tumor, and it can cause brain in pain in brain. Thank you very much.